Hey, welcome to the Biochemistry Academy, where we teach and interpret biochemistry courses for anyone to understand. Now, we began a serial course on nucleic acid chemistry, and today we will talk about RNA, understanding what RNA is, understanding the structure of RNA and the types of RNA. Now, RNA stands for ribonucleic acid, and it transfers genetic information from one part of the cell to the other. Unlike DNA, which is a double-stranded helical structure, RNA is a single-stranded structure. RNA, like DNA, is also made up of three parts, which are a ribose sugar, a nitrogenous base, and a phosphate group. Remember, we said that the sugar in RNA is a ribose sugar, and that is why it is called ribonucleic acid. While in DNA, the sugar is called the oxyribose sugar, and thus it is called the oxyribonucleic acid. Now, what about the bases in RNA? The nitrogenous bases in RNA are adenine, guanine, cytosine, and uracil. So uracil is only found or present in RNA, while in DNA, thymine is only found in DNA. Now we'll talk about it more in our first video on nucleic acid chemistry, so you can go back to understand the difference between RNA and DNA. Well, let us continue. RNA is a linear polymer of ribonucleotides that are joined in a 3' to 5' phosphodiester bond. Now let us talk about the types of RNA. There are three major types of RNA, which are 1. Messenger RNA, 2. Transfer RNA, and 3. Ribosomal RNA. And we should note that all these RNA are involved in protein synthesis, and each differ from the other by function and by their size. Now let's start with the messenger RNA, which is also called mRNA. Now the mRNA comprises of about 5 to 10% of the total cellular RNA. Now it is synthesized in the nucleus as heterogeneous RNA, which are then processed into functional mRNA. Now, these functional mRNA are the one that carries genetic information from the nucleus to the ribosomes, where proteins are made. Now, this genetic information are transferred in the form of codons. So let's understand this structure of mRNA. We can see there is a cap here and there is a tail here, and we see that it runs in a 5' to 3' direction. Now the cap here protects the mRNA from various exonuclease enzymes, and it is capped with the help of 7-methylguanosine triphosphate. Then at the 3' end, it contains a poly A tail, which is a sequence of adenine nucleotides that are added post-transcriptionally. Now, these tails plays a crucial role in mRNA stability, in translation efficiency, and in nuclear export. Now, let us understand the functions of mRNA. Now, mRNA serve as a template for protein biosynthesis, and it transfers genetic information from the DNA, which is in the nucleus, to the protein synthesizing machinery. Now, in eukaryotes, mRNA are monocentronic, meaning that it only codes for one peptide. If mRNA codes for only one peptide, the mRNA is called monosynchronic, and if it codes for two or more different polypeptides, the mRNA is polycistronic. Next, we'll talk about transfer RNA, also known as tRNA, which is a crucial molecule in the process of protein synthesis. Now, the tRNA molecule vary in length from 75 to 95 nucleotides, serving as an adapter with three key regions: the acceptor arm, where amino acid attaches; the anticodon loop, which contains a sequence that pairs with mRNA codon; and the D and T loop, which is involved in proper folding and function. Now, in the structure of tRNA, all single-stranded transfer RNA molecules are folded into a structure that appears like a clover leaf. If we check our diagram here, we can see that the tRNA folds in a structure that looks like a clover leaf. Now, all transfer RNA contains four main arms, which are the acceptor arm, the D arm, the anticodon arm, and the total uridine arm, which is also known as the T arm. Now let us talk about the function of tRNA. Now, tRNA carries amino acids in an activated form to the ribosome for protein synthesis. Next, we will talk about ribosomal RNA, also known as rRNA. Now, as the name suggests, they are the RNAs of the ribosome, meaning that they are the most abundant RNA, around 80% of the RNA. Now, to understand a ribosome is a cytoplasmic nucleoproteins that act as a machinery for the synthesis of protein. Now, it's at the ribosome that is where proteins are being synthesized. We'll talk about this more in protein synthesis. Now, they are composed of two units, a large and a small subunit. Now, these are the subunits in a prokaryotic and eukaryotic cell. Now, this is the normal structure of ribosomal RNA. Now, let us talk about the functions of ribosomal RNA. Now, it is necessary to maintain ribosomal structure, and it also participates in the process of protein synthesis by binding the mRNA to the ribosomes. Now, in recent studies, ribosomal RNA may also provide some catalytic activities and thus an enzyme called ribozyme. There are other nuclear and cytoplasmic RNA. They include heterogeneous RNA, which we say is a precursor to mRNA, and we also have the small cytoplasmic RNA, which it is used to recognize signals for protein synthesis. We also have small nuclear RNA, which function in excision of introns and the splicing of exons. And we also have long non-coding RNA, which have allosteric effects for interacting with different ligand proteins. Well, don't forget to like, share, and subscribe to the Biochemistry Academy for more content like this. Arigato.